So for those of you that don't know me, which is I assume most of you, I'll introduce myself. My name is Chloe. I am a senior at Northeastern University in Boston. I am a behavioral neuroscience major with a minor in economics. And I actually I made this class because I really love genetics. I worked at an epigenetics lab basically all throughout high school, and I've worked in a lab for early research for Alzheimer's, and right now I'm actually working at Massachusetts General Hospital in ALS clinical trials. So if we have time after and you want to ask me questions about any of that, I'll be around. But yeah, so this presentation, if you were just coming from my parasite presentation, this one is rated as more challenging. So I am coming in assuming you have at least some background of genetics, but we are still going to go through everything. But sort of going through the basics, it will be a bit fast. So feel free to let me know any questions you have, any thoughts. I love having this class be interactive, so please put all your questions in the chat, or even feel free to interrupt me and raise your hand, just whatever you feel comfortable doing. And yeah, so let's get started. So before I can actually get into genetics and why it's way more complicated than most people think, first I'll need to give sort of the basic overviews so everybody's on the same page on what genetics is. But first, we're going to talk about evolution, which seems kind of like a weird thing to start for a genetics lecture, but genes are the mechanism of heredity, so I promise it will make sense. But just hear me out. So hopefully all of you have heard of a little guy named Charles Darwin. He published On the Origins of Species in 1859, came up with the idea of Darwinian evolution. And his belief for evolution was that basically you have mutations that occur in reproduction, and those random mutations happen to be either advantageous or disadvantageous. Most of the time they're disadvantageous, but the times when they're advantageous, those mutations will then get passed on to the next generation and the next generation. So Darwin's idea for how giraffes got such long necks is that essentially one random giraffe had a random mutation that made it so that their necks were taller, so they could get to the taller trees. And so they were pretty happy with having this longer neck. And because they were very healthy, they were able to mate. Whereas maybe their shorter their shorter friends would not get a mate. And so then eventually that becomes sort of the dominant trait. And so all giraffes have long necks. Darwinian evolution. But it turns out that before Darwin, there was another guy. And you may have also heard of him, depending on how much you go in depth in your biology class. And this guy is Jean-Baptiste Lamarck. He was French, and actually 50 years before Darwin published On the Origin of Species, Lamarck actually was the one that sort of popularized the idea of evolution. In his published Philosophie Zoologique, which was published in 1809, he proposed his own theory of evolution, again, which was before. This guy inspired Darwin. But his belief was that the way giraffes got such long necks is that throughout their lifetime, they were constantly stretching their necks and, you know, trying to get to the taller trees. And as they were stretching their necks throughout their lifetimes, that then would become acquired, and then they would pass that on to the next generation, and so on and so forth until we get giraffes with really long necks. Now, obviously, Lamarck isn't taught in biology class, right? We are taught about Darwinian evolution, so Darwin pretty much won out. But it turns out, and I think you'll see by the end of this lecture, that Lamarck may have actually, you know, had something going on. I don't know. Yeah. So let's actually talk about genes. They're the blueprint of the cell, right? So genes are made of DNA, deoxyribonucleic acid, which again, as I mentioned, is the mechanism of heredity, right? So the main way that you get your traits is from your DNA. And so your genetic code, if you have a genetic code for brown hair, then you have brown hair. And that is how you look so much like your parents if you are biologically related to them, right? Because the sperm and the egg both have DNA, and they'll sort of combine to create a DNA that is a combination of both the mom and the dad with some random mutations thrown in, and that's you. And one important thing to know about DNA, the reason it's called the blueprint is there's something called the central dogma. So the idea is that we go from DNA into protein, and protein is essentially the building blocks of basically everything in your body. Not everything, but most things. And proteins are made up of amino acids, and proteins do all sorts of things in your body. ATP is a protein. I don't know if you've adenosine triphosphate, which is what is used in mitochondria, all sorts of protein, basically all proteins are made through this process where we go from DNA to protein. But how do we actually get there? Well, we have this thing in the middle called mRNA, also known as messenger RNA, and essentially mRNA is sort of the go-between between getting from DNA into protein. And the process to go from DNA to mRNA, and these are both nucleic acid based, so that's important to note, is something called transcription. And then we translate from the language of nucleic acids into the language of amino acids when we go from mRNA to protein, so that's called translation. So let's actually talk about these. So DNA, sort of chemically, it has a sugar and phosphate backbone. The real information, though, the thing that actually is like, oh, you have brown hair, is carried through nucleic acids or nucleotides. And there are 4 DNA nucleotides. So there's adenine, thymine, guanine, and cytosine. One important thing to note about that is A and T always pair with each other, and so does G and C. And so this is kind of the double helix shape that hopefully we're all familiar with DNA. And then these are actually the chemical structures of the bases. And so let's actually clarify some terms. So the genome is all of our DNA, right? So we have a bunch of DNA that's organized into different chromosomes, and the genome is all of the chromosomes together. It's just the whole thing. Genes are just sections of the genome that go towards making a protein, right? So genes, so the genome is all of the DNA, right? The whole shebang, the whole, I don't know, blueprint workbook, right? And then genes are like, oh, well, this is the blueprint for a chair. And so you can actually see in this diagram on the right, it's pretty cool. It shows chemically, you don't need to understand any of this, but this is like the backbone right here, the orange and the yellow. And then these are the bases. But you see how there are sort of like 2 bonds between the adenine and thymine, and then 3 bonds between the guanine and cytosine. You can almost think of it as kind of like puzzle pieces, right? Or like blocks. Like one block has 3 holes that go out this way, and the other block has 3 like holes where they can fit into. And so a 2-hole block won't work. So that is actually how they manage to pair with each other. It's just because they're soulmates. You know, they want to be together. But that's actually really important because that's how the DNA is able to replicate itself, and it's also how we're able to do transcription. Because what happens in DNA replication and transcription is the DNA gets unzipped. And then what we're able to do is, even though we only have 1 strand, we automatically know what bases should be matching on the other side of it, right? Because they will only pair with the corresponding nucleotide. And so that is actually how you're able to replicate, is because they have this nice matching going on. Yeah, it's not transposition and transcription, it is translation and transcription. But yeah, so how do we actually get from gene to protein. So this is where the mRNA comes in. But first off, why do we even need mRNA, right? Why can't we just go straight from DNA to protein? Well, it's because the DNA is kind of fragile, right? And the DNA needs to be kept safe. So this is a diagram of an animal cell. And so we have the nucleus right here, and that's where the DNA can sort of stay safe. It stays in the nucleus. If it were to come out of the nucleus, the cell is kind of a dangerous place. There are a bunch of enzymes. There are things called lysosomes whose whole job is to go around breaking stuff down. And so if our DNA was just floating around in our cytoplasm, it would get destroyed. So we have to keep it in the nucleus to keep everything safe. But the protein-making factories called ribosomes live out here in the cytoplasm. They need access to the cytoplasm because the cytoplasm is where all the amino acids, all the building materials for proteins live. And so we need mRNA in order to carry the genetic information from the DNA to the ribosomes without hurting the DNA. And so that is what the messenger RNA does. Transcription is the mRNA step. So we have a gene, right, which is again sort of codes for a specific protein, is copied onto a single mRNA strand. So we can take it from the DNA in the nucleus to the ribosome. And the way we do this is another protein, again, proteins run everything in the cell, called RNA polymerase. And RNA polymerase with a bunch of other proteins, red blood cells cannot make new proteins. That is right, exactly. Red blood cells are wild. They carry oxygen. They just have hemoglobin proteins, and that's basically it, and then they die. Yeah, so RNA polymerase essentially will send something called a promoter on a gene. So there's something at the start of the gene to be like, hey, come here, start transcribing at this spot, right? Because we have the whole genome. How does it know where the gene starts? Well, there's something called a promoter region that will attract certain chemical factors, and that will basically recruit RNA polymerase. So then we have the RNA polymerase will come through and add, again, those corresponding bases. The only thing that's a little bit different this time is that RNA is ribose nucleic acid. So instead of using T, it uses U or uracil. So it uses uracil instead of thymine. But other than that, it's basically exactly the same. In fact, the only difference between uracil and thymine is actually just a single hydroxyl group. Basically, one has an extra OH. That's literally the only difference. But yeah, so it's guided by RNA polymerase. We get a nice little group. And then it will actually reach sort of like a termination signal that will tell it, okay, this is the end of the gene. And then we have our mRNA. But wait, we can't actually go to the ribosome yet. We need something called mRNA processing. So mRNA needs some processing before it can be sent out. So there's something called introns and exons. And basically the introns, and I know it seems counterintuitive the way they named it, but I have a way to remember it. So introns stay in the nucleus, so they never leave, but exons get to go out of the nucleus, right? So they get to go out to the ribosome. EX is a thing that means, or is actually the Latin word for to go out of. So that's how I remember it. But essentially the introns get spliced out of our mRNA, and then we add cap and tail, which is really important because remember how I said the cytoplasm is really dangerous? The cap and tail essentially act as like a little thing to be like, please don't eat me. I'm kind of important. Don't break me down just yet. And so that will help protect the mRNA when it goes out into the cytoplasm. But yeah, so now we know how we physically get from gene to protein, right? Because the mRNA then travels out of the nucleus to the ribosomes. So now that we're at the ribosome, we have our mRNA. What happens? Well, mRNA gets translated into amino acids, which is, again, the building blocks of the cell. So every 3 nucleic acids, we transfer to 1 amino acid. So again, with DNA to RNA, it was a 1-to-1 transcription, but in translation, it's 3 to 1. So we have these things called codons. And these codons, each sort of letter, like AUG, each sequence, codes for a different one of 20 amino acids. And yes, the slides will be sent to us, or to you. Yeah. And then we also have stop codons. And the main thing that I want to talk about with the protein code is there's something really cool about it, which is, so I was talking about mutations, obviously, with evolution. Mutations are important. We need to have them in order to grow and change as a species. However, mutations generally, despite what the Marvel comics may have you think, are not good, like 99% of the time. 99% of the time, if you have a mutation, it is not going to help you. You are going to like not be born, essentially, if you have a messed up mutation. Or you have a mutation that will make it so that you will die within 2 months of being alive. So we don't want mutations, generally. So we have a sort of solution in our code to help protect us, which is that there are only 20 amino acids, but there are way more different combinations of these 4 amino acids, or these 4 nucleic acids that can go to these codes. So this serine is 1 amino acid, and all of these different sequences all code for serine. So it's repetitive, not ambiguous, because UCU only codes for serine. It doesn't code for serine and something else. But essentially, let's say that we have, I don't know, UV radiation, right? You're in the sun, and that causes gamma radiation. You get a little bit, and then your UCU gets mutated so that you'd actually go from, this would actually be TC or TCT, right? Because for genetics, but so this would turn into TCC. Let's say we change that 1 nucleotide. It's okay, we're fine. We still have the same end result, right? We still have serine. And so there's kind of like this built-in mechanism to help protect yourself from mutations being deleterious. So that's pretty cool. Yeah, so now let's get to translation and how it actually works. We have this thing called tRNA, or transfer RNA, and it is, on one end of it, it has sort of the corresponding anticodon again. So we're still using the nucleic acids here that have this sort of matching piece to them. And so they are then, on the other side, they're attached to a specific amino acid. And so each one is read as it's sort of read through the ribosome. And then we end up with a peptide, and that peptide will then leave and fold into a protein. And woo, we've gotten all the way from gene to protein in like, I don't know, 15 minutes, less than that. But yeah, so this is sort of like the basic refresher of hopefully what you've learned in biology class. And if you don't understand it, I have a really nice video that's, I think, does a better job than even I do, just because it's visual. And if you're a visual learner, I think you'll really like it. So we're just going to watch that, and then we'll get to some cool stuff. Yeah, let me new share. There we go. And let me know if you can hear it.
The DNA double helix contains 2 linear sequences of the letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make a protein. The blue molecule is unzipping the double helix and copying one of the 2 strands. The yellow chain sneaking out at the top is a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the gene. At this point, the RNA needs to be edited before it can be translated into a protein. This editing process is called splicing, which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons. Splicing begins with assembly of factors at the intron-exon borders, which act as beacons to guide small proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released, and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes remove all introns so that the edited RNA contains only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it sneaks out into the outer part of the cell. Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
So there we go. Going back to the PowerPoint now. Yeah, hopefully, are there any questions? This is a good sort of stopping point before we really get into the meat of the presentation. So what happens to the introns? The introns are not left to float around in the nucleus. There are actually very special enzymes that just break down introns. So they will not hurt any of the DNA, but they will break down the introns. So that's what happens to the introns. Good question. Yeah, so I want to talk about the Human Genome Project. It was started in 1990 and it was finished in 2003. And basically the goal of the Human Genome Project was to sequence the entire human genome. So basically all of our chromosomes sequenced. So this was, millions of base pairs. And it took, 13 years and $2.7 billion, which is a lot of money for a scientific project. But they invested that much money because at the time, they thought that it would revolutionize medicine, right? We would know what caused all of the diseases that affect people, right? Because if we're going off sort of the central dogma of biology, if we know the blueprint, right, then we're good. Like all we need to know is what things in the blueprint can go wrong, and then we've just solved every human disease. So essentially, it sounds kind of crazy now, but they're like, once we have the Human Genome Project, we're going to be able to we're all going to be living to 130. We're going to be surviving way longer, and we're going to cure all the diseases. But obviously, that's not how we're living now. So I want to talk about why the Human Genome Project did not live up to expectations. And this is something actually, Zach or Edward, I think, that you brought up earlier, which is if every cell has the same genes, what makes a muscle cell, for example, different than, let's say, a neuron, right? Or a skin cell or epithelial cell is what they're called. And Edward already said it, but if anybody wants to say or put in the chat any ideas. Just want to make sure y'all are still here. The shape, it is actually not the shape. That is a good guess, though. It depends on what part of the genes are read. Yeah, what part of the genes are expressed, right? So we have the whole genome. Instead of thinking of DNA as like a blueprint, we should think of the genome as like a recipe book, right? And let's say this is the biggest recipe book in the whole world, and it has every recipe for every single food, right? And we can think of your cells as like different kind of restaurants. So like your muscle cell is like an Indian restaurant, and maybe your neuron is like an Italian restaurant or something, right? You don't want to be making, like tacos at an Italian restaurant, right? And so essentially, we actually regulate sort of what recipes are used by regulating the expression of the DNA. And so DNA is expressed differently in different cells, depending on what sort of function they serve. And there are two main ways that this happens. There's methylation and histone modification. Now, they do seem kind of complicated, right? They're complicated words, but I promise I'm going to make it really simple. So let's say we have our recipe, right? So let's say this is a gene, but we're going with the recipe metaphor here. This is Caesar salad, right? And we've got the list of ingredients, right? So we can imagine these as, amino acids, right? So these are like these would be 3-letter codons. If we methylate this gene, methyl group is literally a chemical CH3 group that goes on top of the nucleotides and blocks the RNA polymerase from attaching. So this is methylation. Can you read the recipe? No, right? So that, can you make Caesar salad? No, you can't, because you don't have the recipe anymore. So Caesar salad doesn't get made, or the protein that you want to make doesn't get made. The other way that it happens is histone modification. And histone modification is basically sort of like the structure of the DNA. So the DNA is in chromosomes. It's actually not in chromosomes except for when it replicates, but that's for a later time. But basically, the DNA is wrapped around histones, and it's sort of like a spool thread kind of thing. So you can imagine like thread wrapped around a spool, and that's how DNA is wrapped around these histones. And based on sort of how tightly it's wrapped, it will basically physically block the DNA from getting accessed by the RNA polymerase, which needs to happen for it to be transcribed. And so the way to imagine this, again with our recipe, so methylation is covering it up, right? Histone modification and acetylation is essentially, let's say we put some histones, acetyl groups on it. Can you read the recipe? No, right? Because it's crumpled up. So essentially, these histone acetyl groups will make the DNA really tightly coiled, and so you can't get to it. But there is an enzyme called histone deacetylase that will sort of unwrap the protein, and so it will get rid of the acetyl group, so we can unwrap it, and so then we can make our recipe again. But these are the main mechanisms of epigenetic modification. So there's histone acetylation and methylation. So now you know. So let's talk about what this actually means, right? Because right now we've been really, I feel like, theoretical. So let's actually get into what this looks like in real life. So here we have these agouti mice, which is probably the most famous example in science of epigenetics. So these mice look pretty different, right? There's something going on here. But what if I told you that these mice were clones? These mice are genetically identical, and these mice were gestated in the exact same mother. Pretty crazy, right? You're like, so why are they so different? Well, not mutation. There was no mutation involved. That's the crazy part. It actually has to do with epigenetic modification, which is what we've just been talking about. So essentially, they bred these mice. This is mice bred in a lab to have a gene that they call the agouti gene, because the scientist that made the gene was like agouti. That was his name. But so this agouti gene, if it's expressed, and in most mice it is, if it's there, then it causes hypopigmentation, aka this sort of orange color, right, instead of the normal brown. And it also causes a slower body metabolism, therefore leading to obesity. So obviously you can see this mouse is a lot bigger than the mouse on the right. But this mouse on the right still has the agouti gene. So I'll tell you what's different about these two mice. So even though they were gestated in the same mother, the mother was fed two different diets. So when she was gestating this mouse, she was fed a sort of just like a normal mouse diet, right? Whatever they give in the lab, lab rat food. But the mouse on the right, the mother mouse, when she was gestating this brown mouse, was given B12 vitamins and folic acid. And you're like, okay, so what does that have to do with anything? Well, B12 vitamins actually have methyl groups. So they were actually donating, by having this supplement in the mouse's diet, they had more methyl groups available. And so the scientists actually found that these, the B12 then donated its methyl group to methylate this gene. And so now this gene isn't getting expressed in this mice, so it grows up to as if it doesn't even have the gene. That's pretty crazy, right? But wait, it gets even more crazy, right? Because we're talking about mice. It's like, meh, mice. We use mice because they're experimentally valuable, because they're similar to humans, but you know, they live a shorter lifespan, right? But they're not people. And it's also nice because we can kind of torture them and do awful things to them and give them cancer. And, you know, we won't get in trouble with any ethics boards, which is why I've never worked with mice, but like, you know, some people have the stomach for it. But so it's obviously hard to do sort of an experimental procedure like they did with the mice and people. But we do have some examples from history that have sort of led to kind of the perfect experimental conditions, unfortunately. And so one very clear example of this, and perhaps the most famous, is the Dutch winter famine, which occurred in World War II. So basically, for a period of a year, essentially, the Dutch winter famine was this blockade where Hitler was like, all right, you guys don't get any food. We're not going to let you get any rations from other countries. And they actually had to be like airdropped in, but it wasn't enough. And so this is actually a picture of people. These are food bags, but that was the only food they could get because they also like raised their fields so they couldn't grow anything. But yeah, so basically everybody there was malnourished and starving, which is not great. And they found something really interesting, sort of many, many years later, which is they were looking at people that were born specifically just after the Dutch winter famine. So people who were in their mother's wombs during the Dutch winter famine. And they found something really strange, which was that they were much more likely to, one, have glucose intolerance, aka develop type 2 diabetes. They were also much likelier to have a higher BMI, so more prone to obesity. And they also had a much lower lifespan in general. So they died a lot younger than their siblings. Oh, is Birch here? Oh yeah, this is something to lighten the mood.
I'm unhappy right
Yeah,
now.
it's my kitten. But anyway, so as I was explaining, they have all of these differences to their siblings. So they're like, what is going on? And yet all these people have it. And they didn't even live through the Dutch winter famine, right? They were born after it. So what's happening? Well, they actually looked through their genes, and not just their genes, they looked through their epigenome. And what they found was that in this group, there was a certain gene called PIM3, which had been demethylated. And so normally, this gene is methylated and it's not expressed. But when this gene is expressed, it slows down the body's metabolism. And you can think about, like, imagine you are a baby that is gestating in a mother that is severely malnourished, right? It would be advantageous to you to have a slower metabolism, right? Because if you're growing up in a famine, you do not want to burn through your energy really quickly or you're going to die. And so you can see how there's this literally like acquired trait while they're in the womb of this demethylation of this gene that then leads to an entirely different kind of lifespan because they then grew up in sort of normal conditions, normal food conditions, where having a slow metabolism is not beneficial. And so then they developed type 2 diabetes and had all of these terrible health effects. But we are actually seeing sort of how epigenetics, again, is causing these lasting changes. And the weird part is, it didn't just happen to them. These effects carried on to their children. So the methylation or demethylation of genes is actually something that can be transferred to the next generation as well, which is really crazy. And also sort of really, it's kind of proof, if you think about it from social justice issues and sort of cycles of generational trauma, there is literal biological trauma that happens in changes to the expression of our genome. So you know how the B12 supplements in the mice led to sort of normal methylation can occur if you're malnourished. And so, right, these fetuses were not getting enough nutrients from the mother. Oh yeah, one other really weird and awful thing that I should mention. They did another experiment with these mice, by the way, where they fed the mother mouse the B12 vitamins, and then they also fed the mouse BPA in the water, basically to the levels that we have in our water. BPA is something that is found in some water bottles. And it completely canceled out the positive effects of the B12 vitamins because it essentially sort of stole the methyl groups. So, you know, pollution, woo! We're living, like, we have microplastics in our bodies right now, and that is potentially permanently affecting our epigenetic makeup. Woo! Love that. Did later children of those mothers also have slower metabolism? No, they didn't. They didn't. So late children that had sort of before and after normal metabolisms. But it was just the children that were gestating in the period of the Dutch winter famine that had that demethylation happen for their embryo. Yeah, but it's crazy. So we're talking about epigenetics, and we've talked a lot about fetal development. But what about when you're actually born, right? Well, they've done studies on that too. So there's something called CREB, which is a protein that is involved in sort of the brain and regulating all sorts of sort of brain processes and neuroplasticity, and mouse mothering. So one thing you need to know about mice is the mouse mothers show affection to their children by licking. It's like getting a nice warm hug from your mom, but the way mice do it is licking. So if baby mice don't get licked, they're not in a good place. They've been abandoned, right? Because that's the signal to them that they are getting loved and they are cared for by their mother. And scientists have found something really strange, which is that the baby mice that don't get licked are more fearful when they grow up, which I guess makes sense from like a behavioral standpoint, right? But they also found that they literally release more cortisol, like physiologically, and less glucocorticoid in response to stress, which is, you know, really strange. What is changing physiologically there? How is that happening? And so they have this idea that getting licked protects mice from stress, and when you're not licked, then you have more stress. So what's going on there? Well, it's a very complicated process, but let's say that you're a baby mouse and you're getting licked, you're having good, happy feelings, right? So your serotonin receptors start getting activated because your brain is releasing serotonin because it's happy and it's in a good mood. This activates the Krebs cellular cascade. So essentially this Krebs protein that increases neuroplasticity starts activating all of these different cellular proteins, and we have this wonderful cascade of crazy things. And again, proteins do a bunch of stuff in your body. This will then activate the expression of nerve growth factor, actually through demethylating the nerve growth factor gene. This nerve growth factor then will demethylate the glucocorticoid receptor in your brain. And so when that is demethylated, you express more glucocorticoid. And glucocorticoid helps you deal with stress. And so because this nerve growth factor is demethylating the glucocorticoid receptor, we then have this epigenetic change where the genes that are being expressed in your brain change so that you have more expression of a hormone that helps you deal with stress. And so obviously the mice that don't get licked, they don't have that cascade, so they have less glucocorticoid receptors, so they're less able to deal with stress. But now that we've talked about all of this epigenetics, right, methylation and acetylation, the Human Genome Project seems like, you know, it might be missing some things, right? Because it's not just enough to have the DNA sequence, right? We need to know how it's being expressed in different cells and in different people. And so the Human Genome Project doesn't cover any of that. But there's something even crazier about the Human Genome Project, which is there are around 400,000 proteins in your body, like unique different proteins. So there are way more proteins in your body, but there are 400,000 unique proteins. That's a lot, right? So do you have any guesses for how many protein coding genes there might be in the human genome? Please put in the chat what you think. Any guess is fine, really. 90 million, 5. These are wonderful guesses. I love it. 1.2 million, 80,000, 800,000. So the estimate that scientists had, 2.9 billion, that's a lot. These are all wonderful guesses. I love it. But the estimate that scientists had, and don't worry, the scientists were wrong too, so don't feel bad about getting it wrong. But they believed that there would be about 100,000 protein coding genes, right? Because they're like, well, some proteins are just like 3 proteins sort of linked together. So like, obviously, some of them are just proteins being built together like post-translationally. And so they're like, all right, there's probably 100,000 protein coding genes because obviously there are millions of base pairs, so we've got to have a lot of genes, right? A lot of protein. But no, 25,000. So I guess it's closer. I guess, Zachary, you were correct. You were the closest of anyone to the correct answer. But yeah, so the actual number was 25,000. And when scientists discovered this, they were like, what? This doesn't make any sense, right? Because how can there be 400,000 different unique proteins in the body and only 25,000 protein coding genes? It's just like, does not compute. So here's why. Remember how we were talking about mRNA processing and introns and exons? Didn't you think it's a little weird? It's like, how does it know what's the introns and what's the exons? Well, it turns out that it kind of doesn't. So mRNA, again, we have the introns, the exons, and we have spliceosomes that splice out the introns. But it turns out that there's this thing that's actually very common called alternative splicing, right? So we have maybe like traditional splicing for that gene, right, where we cut out certain introns and then we get our mature mRNA. But what if we just cut here and here and then we forget to cut here at this spot? I don't know if you can see my mouse. You probably can't. But what if we just forget this exon in the middle and so it doesn't get added? Oh, okay, great. Yeah, so what if we don't cut here and here and then this just gets considered an intron? Now we have a totally different mature mRNA, right? So now you can see how we can get from one gene to a ton of different variations of proteins. There are some genes, in fact, that we've discovered that have over 200 different alternative splices. And so you can almost think of this, again, going back to our recipe, right? So if we have our recipe, let's say, oh, well, I want a Caesar salad, but I want it without Parmesan cheese. That's going to be a different salad, right? Or, oh, I want a Caesar salad, but without the lettuce. That's going to taste very different than a normal Caesar salad, right? So now you can sort of see how we get from so few protein coding genes to all the diversity of proteins that we have in our body. But yeah, it gets even crazier than that, right? So we have, how many protein coding genes again? It's like 25,000. So what is the rest of the DNA? Because our genome is huge. Well, turns out that 95% of our genome is non-protein coding DNA. Which means the whole thing that you learned in biology about DNA to mRNA to protein, the central dogma that we've just been going through, only 5% of DNA actually does that. So what is the rest doing? Well, scientists were like, we don't know. It's junk. They called it junk DNA for years. They were like, we don't know what it does, so we're just going to say that it's junk. I guess it's just there. But we've made some inroads since then. So we now realize that a lot of these trash DNA are filled with things called transposons and retrotransposons, or jumping genes. And they were first found in corn because they're actually 85% of the corn genome. And transposons, the scientist was looking through the corn genome, and they just found these little sections of DNA that were like a single sequence, and that they were sort of like repeating all over the genome in the protein coding regions and the non-protein coding regions. It's just like, you know, like getting glitter in your carpet, right? You can never get it out, and it's just everywhere. That's retrotransposons. And so it's just sprinkled all throughout the genome. And what we found is that these DNA strands can actually self-replicate. So they kind of do the first step. So retrotransposons kind of do the first step. So they do transcription, but instead of getting translated into protein and leaving the cell, they actually do something called reverse transcription, where they get turned back into DNA, and then they just insert themselves randomly all over your genome. And so that's why they're called jumping genes, because they're jumping all around your genome. And so most of the time, these transposons will go into non-coding regions of your DNA, so you're probably fine. But let's say that we get a transposable element inserted into a part of your DNA that codes for a protein. Congratulations, you now have a changed protein, right? It has a huge mutation in it. And these transposons and retrotransposon changes can actually happen throughout your lifetime and then be passed on to the next generation. But yeah, so we have also DNA transposons. And so we have DNA intermediate and target DNA. And in that case, it literally just replicates a form of DNA and it just inserts it somewhere else. So it's less steps than the retrotransposons. But yeah, so how does this actually, oh wait, okay. So one important thing to note, obviously transposons seem like they would be pretty bad, right? If they were constantly active in your body. In fact, there are no longer any active human transposons. We do have some retrotransposons, but that's about it. And generally you don't want transposons to be active because they're gonna cause a bunch of changes to your DNA. They're like, you know, mutation lottery machine, and like 99% of the time you're gonna get a bad mutation. So generally, retrotransposons and transposons are heavily methylated in the DNA. They're heavily methylated, they're heavily acetylated, again, so we're talking about they're really sort of crumpled up, right? So they can't go out and escape and replicate themselves. They can't be transcribed. Generally, that's how it works. Unless we found that actually something really crazy happens when animals are put under stress. So remember our mouse mothering, right? Getting licked. Getting licked is pretty good. You know, you're in a good place. But let's say you're not getting licked. Not getting licked actually causes the demethylation of line 1 retrotransposons. So basically what happens is your line 1 retrotransposons become super active and they start inserting themselves all over the genome. So this also put all of these baby mice that were more stressed at more risk for health problems and also their future generations. And the really interesting part is you can almost think of it as like an evolutionary advantage. By changing the rate, you can have some control over changing the rate of your own mutations based on your environmental situation, right? And so that's actually what's happening a lot in climate change. What I studied was plants, and plants have the same kind of epigenetic mechanisms with transposons. And they found that when the plants were experiencing environmental stresses, they demethylated their transposons and the transposons start going crazy. And essentially you can think about how this is beneficial. You're like, well, my life is already pretty crap. I'm gonna probably die because my mom doesn't love me. So, you know, might as well just, you know, try the lottery and hope that I get something good because that's all I can do. And so this has so many implications in terms of, you know, what we do in the real world. So yeah, what about the rest of the non-coding genes? Because I said 65% of the human genome is transposons, right? And then we have 5% that's protein coding genes. So what's the rest? Well, scientists again were like, oh, this is junk DNA again. But no, it's not. It's not. We're discovering that it actually is able to regulate a lot of really, like all of the things that we've been talking about. So like alternative splicing, what causes a gene to, or what causes mRNA to be spliced in a certain way can be a long non-coding RNA that'll essentially like block the spliceosome from getting to a certain site. And then we have long non-coding RNA that can act as sort of like a dummy for transcription factors so that another gene doesn't actually get transcribed because the promoter doesn't, because the polymerase never gets recruited. And then we also have long coding RNA that can come in and actually change the acetylation and methylation of different genes. And so now you can see, you know, you've learned in biology that DNA is like this, you know, blueprint. It's this passive thing, but no, it is alive and it is, you know, constantly regulating itself. It is not the blueprint. It is like part of the architectural team through this long non-coding RNA. That again, it never turns into a protein, but this is actually what a large part of your DNA does. And there's one other really cool thing that I'd like to talk about, which is post-transcriptional regulation. So remember, we're going back to our transcription translation, we have mRNA. So there's actually this thing where non-protein coding DNA can get transcribed into RNA. This RNA isn't ever going to be turned into a protein, but it has a certain matching sequence with another mRNA, and it'll bind to it, and it'll literally block that mRNA from getting translated, so you don't get the protein anymore. And so this is actually, it's a really complicated diagram, but essentially this is like the little, it's called a microRNA or siRNA, short interfering RNA, but it can come in here and it will attach to the mRNA and prevent it from being translated. That's what this like flat thing means, is that it's stopped. And then it also will signal this mRNA to then be degraded. So the cap and tail doesn't do it any good. So it just never gets turned into a protein. And what's really cool is that there are lots of drugs and new pharmaceuticals that are actually taking advantage of post-transcriptional regulation, and they are using microRNAs to stop the translation of proteins that are dangerous. And so a lot of pharmaceuticals are actually using microRNA, which is really cool. But yeah, so that's kind of like the future. There's still so much that we don't understand about genetics and epigenetics and even proteomics. There's a whole thing that I didn't even get to talk about with, you know, like protein degradation, that like ubiquitination, that's awesome, but out of the scope, but it's super cool. But there's just so much going on in genetics right now, and it has almost nothing to do with, you know, the central dogma of DNA, mRNA, protein. So what does this mean for genetics, and what does this mean for you? Well, if somebody tells you that there is a gene that is associated with a disease, that doesn't equal destiny, right? So take any gene, you know, with a grain of salt. Just because you have a gene associated with a disease doesn't mean that it's expressed. There are tons of environmental factors that can potentially change that. And epigenetics is reversible. And lifestyle and psychopharmacological changes can actually change your expression and even your genes in terms of retrotransposons. And so I think the main lesson that I take away from learning all this is take care of yourself. Self-care is important, not only for you, but any potential biological children you may have, because it doesn't just affect you that day. It may have long-lasting effect on your epigenetic makeup and what you pass on to the next generation. And so I think it's a good explanation. You know, if you're ever like need to take a mental health day, you can just tell your parents, well, I'm just, you know, keeping up my epigenetic fitness, you know. But yeah, it also bodes very poorly for the world that we're moving into with industrialization and everything. And also it really shows how there are sort of biological scars of, you know, years of discrimination and cycles of generational trauma, that even if you're not experiencing it behaviorally or yourself, you're still dealing with the biological effects, because that's still seen in your epigenetic makeup. But yeah, with that, how much time do we have left? Am I over time? Okay, great. So that's plenty of time for questions. I hope you guys enjoyed Splash. I know this was the last class, so you're probably all a little bit dead.